Alright, this is probably a review for you at this point, but remember that when a gene is transcribed, it first contains introns and exons. Which part is the code for the protein? Right, the exons. And the introns get spliced out and left in the nucleus to be degraded. But this brings us to another cool tool that the cell can use to regulate gene expression, which is called alternative splicing. So what's alternative splicing? So you've got all these exons, but you don't need to use all of them when you splice them together. Exons can be combined in different combinations to create different versions of the protein, and exons often correspond to specific protein domains. For example, maybe there's a protein that has a DNA binding domain, a catalytic domain, and 2 regulatory domains, so it can be repressed by other proteins. If for some reason a cell only wanted one of these regulatory domains to be present in certain situations, it can do that by keeping or removing that exon in the mRNA. This saves a lot of space in the genome compared to having 2 entire separate genes, which is especially useful for viruses since they have a very limited space for their genome. Alternative splicing is another area in which mutations can cause disease, though, with a key example being beta thalassemia. In beta thalassemia, a gene that codes for the beta-globin protein in hemoglobin has a mutation in part of the gene that normally allows the spliceosome to recognize the intron exon